degree in he did his master's as a natural science in molecular biology and molecular genetics in Austria. And then he did his PhD as well in Vienna, Austria, and came to the United States for his postdoc, and then rise through his career now to become associate professor as part of the Department of Chemistry and Biochemistry at Montana State University. When prompted what piqued his curiosity for his interest in science, especially in Archaea, he said in high school, he read many popular science on quantum physics, astronomy, astrobiology, and also a lot of famous scientists. And then when he listened to a lecture on microecology, piqued his interest. And after doing his master's, he was asked, to join the lab of his mentor. But he did mention he would join on the condition that he's gonna work on Archaea because he found it very fascinating. And what energized him about his research is that to see what no one else, and ideally think what no one else has thought. And his hobby is playing board games. And a special announcement for everyone: Roland is looking for a new grad student to start next fall, and also looking for a postdoc to join his lab to work on methanogens and other Archaea in the Yellowstone hot spring and the deep sea. So without further ado, Roland, the stage is yours. So just for the bench. Right.
All right, thank you very much for the introduction, and thanks everyone for joining so early. All right, so as a quick reminder, what methanogenesis is. I think most of you are familiar with that, but as a reminder, so methanogens are anaerobic microbes that conserve energy coupled to methane production, which is a trait, as far as we know, that is unique to the Archaea. And these methanogens are responsible for, give or take, 60% or so of global methane emissions. The vast majority of other emissions is coming from abiotic sources. The problem with that is that methane is a very climate active gas with a greenhouse warming potential about 28 or so times higher than carbon dioxide. And the sources of that methane ultimately are CO2 and some low molecular weight organic carbon sources like acetate, formate, and different methylated and methoxylated compounds. And it is pretty much textbook knowledge these days that, and for a long time, that all known methanogens are members of this now superphylum Euryarchaeota that was already described by Carl Woese originally, when the archaeal domain was split into Euryarchaeota and Craniarchaeota in the early days. And so there's several reasons other than the global warming potential that methane is important. The first one is from an evolutionary perspective. We understand that methanogenesis was among the most ancient metabolisms to evolve. It is stated to be somewhere in the range of 3.2 to 3.4 billion years. And there's evidence both from molecular clock analysis, but also more importantly, from isotopic and geological evidence in the rock record. So the current hypothesis is that most likely the last common ancestor of all extant Archaea probably was methanogenic. And because methane has been detected on other celestial bodies, like for example in ppb concentration on Mars, but also on the plumes of Enceladus, anaerobic methane cycling, biological and abiotically, has important implications for astrobiology. So there's a wide diversity of methanogenic pathways that differ depending on what the respective substrate is that these organisms start with, but all these pathways converge on the last step that is catalyzed by methyl coenzyme M reductase, or MCR. It's the only abbreviation I'm going to use for today, that catalyzes the last step, which is the actual release of methane from these organisms by basically cleaning this methyl group of coenzyme M and then reducing it to methane, which is a exergonic process that occurs at about -30 kilojoule per mole. So while the individual enzymes in organisms differ, they all converge on this one step. And this is why methyl coenzyme M reductase genes have been used as a metabolic marker gene to screen environmental samples, and these days mostly metagenome assembled genomes, for the potential of organisms to be involved in. Folks, can you guys mute yourself? Thanks. To be involved in anaerobic methane cycling. And so what we know today is that all confirmed organisms that are involved in either anaerobic methanotrophy that use similar enzyme complexes, as well as methanogens, fall within the superphylum Euryarchaeota. But in the last 10 years or so, several lineages inside the Euryarchaeota and outside the Euryarchaeota, most importantly members of these TAC superphylum. There's several lineages within the Archaea and also others have been implicated by these gene surveys or metagenome assembled genome predictions to be involved in methanogenesis. But the problem is that until very recently, basically our work, no one has experimentally shown that these organisms actually are involved in anaerobic methane cycling. So while I today gonna focus solely on members outside the Euryarchaeota, I want to highlight that we also work on new lineages within the Euryarchaeota that are involved in methane cycling. If you're interested, we have a paper in review that is on bioRxiv that was done by my fantastic graduate student, now PhD, Mackenzie Lyons, who also co-organizes the Archaeal Power Hour, in which we study methanogenic archaeoglobi. But so for the talk today, we're going to focus on members of this TAC superphylum, so members of the Thaumachaeota, Eigarchaeota, Crenochaeota, and Corachaeota. And so this study started by the results from a recent study by Mackenzie and my former postdoc, Dr. Viola Krugenberg, in which they conducted mesocosm experiments, so mesocosms that were seeded with material from 3 hot springs in Yellowstone National Park that were incubated in the presence of different methanogenic substrates or precursor substrates, and then observed production of methane and the microbial community composition over time. And the result of this was that we identified specific conditions under which different potentially methanogenic organisms, so MCR encoding Archaea, thrive. And we use these incubation setups to basically formulate cultivation conditions and media to actually bring these organisms into culture. And so the heroes of these stories are really my awesome graduate student, Anthony Coates, my former postdoc, Dr. Viola Krugenberg, as well as my staff scientist who is doing a lot of bioinformatics in my lab, Dr. Zachary J., which resulted in 2 papers that are currently in revision and in which these 3 are co-authors. So the first one is a methanogenic coracheon that Viola was able to culture from a hot spring in Yellowstone National Park that runs methanogenesis using methanol as its only substrate, in which we, over hundreds of days, about 2.5 years now, that we have this culture growing, produces substantial amounts of methane, up to 4% or so in the headspace, from methanol that grows at about 77 degrees centigrade, about circumneutral pH, and there's no other methanogens in this culture. It is about 84% enriched, and there are these typical, very awesome, beautiful, large filaments of Corachaeota, sometimes growing 20, 30 micrometers in size. And you see the number of these Corachaeota cells in red here as the proportion of the total microbial community reaches about 80% or so of the total microbial community over the growth of this organism, shown here as the percent methane in the headspace. We demonstrated the methanogenic capabilities by a combination of genome sequencing, obviously, as well as stable isotope tracing, in which we showed a conversion of C13 methanol to C13 methane in the culture. If you inhibit the culture by BES, a known inhibitor of MCR, you immediately shut off methanogenesis. And if you combine this with single cell activity tracing, this is a combination of cardfish, which we use to identify our coracheal methanogen, as well as SpawnCat, which is a technique that labels translationally active cells. You see that under methanogenic conditions, basically all the Corachaeota in red are also translationally active, but under non-methanogenic conditions, so if you inhibit the culture while the Corachaeota are still present, obviously because they've been grown up earlier, they virtually completely stop their translational activity, linking on a single cell level the dependency of these organisms on the MCR enzyme and linking them to the methanogenesis potential. We've obtained the circular genome of this organism using a combination of long and short read genome sequencing and shown that it is a very close relative of a previously discovered metagenome assembled genome that was discovered by a researcher, Luke McKay, who now works for a private company, who at that time was also at MSU, and showed that these organisms are very closely related. And one interesting thing about these organisms is that they have the genomic potential, yet experimentally unproven, however, that they might be doing things other than methanogenesis. So this is one of the things that we're going to work on in the future to see under which conditions these organisms run methanogenesis or potentially other metabolisms. The second organisms we brought into culture, which was spearheaded by my graduate student Anthony Coates, is a member of the Methanosuranting cola. You might be more familiar with these organisms by their original name, Euryarchaeota or Euryarchaea. We have to rename these organisms, unfortunately, now. We can go into details if you want later why that is. The preprint for this paper is on bioRxiv if you want to give it a read. But in short, these organisms run the same type of metabolism. They convert methanol to methane, and they reach about 40% or so of total cells as quantified by cardfish. The culture grows really dense, which is awesome because that allows us to, in the future, do studies on some of the proteins of these organisms. We have enough biomass to actually extract proteins and do biochemistry studies. And this is one thing Anthony wants to do in his future independent research. So they form these really beautiful small cells that make up 40–50% of all the cells in our culture. We also have a circular genome, about 1.5 megabases, again, obtained by a combination of long read and short read sequencing. And just as the Euryarchaeota, they also encode the potential for alternative growth strategies, which is one of, for me, the most interesting question about these organisms. Under which conditions do they run methanogenesis, and under which conditions do they not contribute to methane cycling? One thing, however, we did that we did not do for the Euryarchaeota, at least not yet, was to look a little bit deep into the cell biology of these organisms. And we teamed up with the lab of Dr. Martin Pilhofer at ETH in Zurich to perform cryo-electron microscopy studies to study the organisms a little bit more in detail. For example, we could visualize there's at least one, possibly two different types of virus-like particles inside of the cells. These red dots represent the individual ribosomes inside of the cells. They have chemotaxis areas that are anchored at the Archaea that are responsible for locomotion. And maybe most intriguingly, these cells also have these, what is shown here in green, unknown proteins that are quasi-crystalline protein structures inside of the cell. We have no idea what these things are doing inside of the cell, but they make up a large proportion of the biovolume of all the cells that we have so far studied. The other thing that is really intriguing about these organisms is that they can form cell-to-cell junctions or cell-to-cell bridges in which there is a shared cytoplasmic space between two or sometimes even three cells. And in at least one instance, we also see electron-dense material being exchanged between these cells. So not only are there cell-to-cell bridges that are about 200 nanometers thick, they share an S layer, they share cytoplasmic membrane, but we also see structures reminiscent of ribosomes and some unknown filaments that are being exchanged between cells. And this might have implication for gene transfer between these organisms or some type of yet uncharacterized metabolic interactions. So with that, I hope I could show you that the power of cultivation lies into actually experimentally proving that vast amount of hypothesis that is pouring out from metagenomics, and that we can see metagenomics as a starting point, but not an end point of the characterization of the physiology of organisms. We have cultured the first methanogens from outside the Euryarchaeota, and we can say now that at least 3 phyla, Euryarchaeota, Methanoceranticola, formerly Archaea, and Euryarchaeota are involved in methanogenesis. Some of these organisms have a very complex cell structure, and up next we will study under which conditions these organisms will perform methanogenesis versus other growth strategies, to try to put the results from pure culture into environmental perspective, to see if these organisms actually live mainly by methanogenesis or other means in the environment. And I can also hint towards that we have more cultures that are coming soon. And with that, I'm just gonna remind, I'm looking for a new postdoc to start at some point next year, the sooner the better, and a new graduate student to start in fall next year to continue work on these awesome organisms using the variety of tools that my lab is using to study these organisms. And with that, thanks for your attention, and I'm happy to address any questions you might have.
Okay, so let's all thank to Roland for this great talk. And if anybody has questions, please, I invite you to either choose the raise hand function and we can let it to you so you can ask your question directly, or either you can put your questions in the chat. Okay, let's go into Brendan.
For me, it was just an applause. I thought it was a very nice talk.
Is that,
so thank you so much for giving a talk at such an early hour.
Thank you.
Anybody?
I did have a question. If you had caught any of your potentially viral genomes in your sequencing.
Yeah, we have a lot of virus-like sequences, but as far as I know from the recent update, we do not have like complete viral genomes or something like that. And this is, at least at the moment, not something we're actively working on. So basically, this only popped out basically because we saw this organism or these viruses or virus-like particles actively infecting a lot of these cells, so to speak. But at the moment, we are not working actively on it, simply because we need to pick our battles. And there's a lot of things that are more interesting at the moment than the virus-like particles. But we have started to talk with people to work on this collaboratively in the future.
In your longer term cultivation, do you see sort of like die-offs that might indicate sort of lysis infection?
Yeah, we actually do. We do this for the Archaeota. Give me 1 second. I think you might actually, I'm not sure if you see this in the growth curve that I showed you, but yeah, you do see. So green, right, is the relative proportion of a target organism. And you see that once they reach stat phase, they actually go down. So that is the only thing that we know that is consistent with the idea that there is viral predation going on. However, I base this only on the fact that we see virus-like particles inside of the cells that are entering and exiting the cells, and that this is coinciding with in the stat phase cells going down. So there's no direct evidence whatsoever. And I stress that no one should make any conclusion of that just based on sequencing data.
Yeah.
You simply cannot do that. You need microscopy.
Okay, we have another question. Katharina, please.
Yeah, thanks, Roland. It was a great talk.
Thank you.
Both of the cultivations that you did, they were growing on methanol. How is because there's always the question about the environmental relevance of it. Like, how do they get like in the environment? Where does the methanol come from? Is it a prominent compound?
Yes, that's an awesome question. We don't know. So to point out, so we tested different methylated substrates. They do not use any of them. They only use methanol, as far as we can tell. And the problem is, and this is where this perspective from the fieldwork comes in, right? Some of the, or most of the hot springs that we study, where we know these organisms are abundant, they are over the boiling point of methanol. So that doesn't mean that there's not methanol around, right? It might just immediately go into gas phase, but that raises the question whether or not these organisms actually grow by methanogenesis in the environment. And this is why I do not trust genomic predictions in general, right? Where I think you always need to test this, because even though these organisms might be doing methanogenesis in the lab, it might not be doing it in the environment because the substrate might simply not be around. So there are sources of methanol because there's bacteria, right? There are presents that degrade like plant organic matter, for example, that is around the hot spring. So I think there are sources of methanol. However, we do not know how stable, quote unquote, the methanol is in the environment. We simply don't know yet. And so far, we have no way to track methanol in these very hot environments directly. Like, we're working on it, but yeah, we are not sure yet. But so this is why I think there's a good chance that these organisms might not be living by methanogenesis in situ, which would have very important implication, right? Because again, this means that even if you detect a known methanogen in the environment, it doesn't mean that they run methanogenesis.
But it would be cool to see what they then actually do in the environment. I'm
so based on
looking forward for your follow-up studies.
Thank you. So based on genomic predictions, they do in one case either dissimilatory sulfide reduction or anaerobic respiration, and in the other case, the methanoserantin cola can do basically fermentation of amino acids. There might be other options, but that's the ones that are the most obvious from the genomic reconstructions.
Great, thank you.
Thanks all.
We have another question, Sebastian.
Yeah, thank you, Roland, for the talk. It's really nice, and I really was puzzled by the trio ET that you were showing. And I'm wondering, because you don't have a pure culture, if I got it correctly. So, do you know between which type of cells the connections are probably done? So
yeah, we do.
Okay.
Yeah. So basically, it is done indirectly, but so the fact that the cells that we're observing here are the only coccoid-shaped cells with an S layer basically means that we're looking at a target organism. So we basically are just lucky. They are the only coccoid cells with an S layer, basically. So it's done indirectly. That being said, is we are working with Martin to try to do correlative fluorescence and electron microscopy, which is, however, very cutting edge. Personally, I don't think we need it. Because I think it is an indirect demonstration, but I think it's very clear these are a target organism. And we only see cell-cell connection between self. so between methanosurrantin cola with
Okay,
itself.
that was
We have
basically the
not
question.
seen any indications that they do it with any other cells. So in this particular image here, right, to fast forward, all these two cells are very clearly methanosurrantin cola, and this we believe is a, in lack of a better term, a swollen up methanosurrantin cola. Maybe it's close to lysine, but it's still the only coccoid cell with an S layer, basically. So indirectly demonstrating that this is also a target organism. So we have in total, like, I think I want to say 54 or 52 cells where we see these cell-to-cell bridges. This is just the most striking example, which is the only one where we see this between 3 cells rather than just 2. But so I have here some other images somewhere. Yeah. I don't. Yeah.
No problem. Thank you. I understand now.
I think,
Thank you.
sorry, I think we have one more question, so we can move on to the breakout rooms. Nicole?
Thanks, great talk. So I'm curious about some of the, ordered structures that you've seen, and you kind of invoke viruses, and it's hard to tell from these, just a few short slides, but it can be difficult to tell the difference between viruses and things like metabolosomes, right? And so there are lots of organisms that have, pretty complex biochemical structures that are not viruses, but are, probably evolutionarily related to viral capsid, things like that. So have you looked, is there a homology to any of those types of, what's been identified in bacteria as metabolosomes or other biochemical structures?
So I don't think we've done that. I think Anthony's online. He can chime in if he is looked into that. So this is why I stress. So I've labeled it as viruses. This is not how it's labeled in the paper. It's labeled as virus-like particles. I simply wanted to reduce the text, but this is why I stressed these are virus-like particles, and unless we have direct proof that these are viruses, I wouldn't call them viruses. So you are right. I should not label them viruses. The reason is I wanted to save space. But yes, so this is one of 2 or 3 different types that we see. And we have several reconstructions, however, where we see this either being attached to the S layer or to the membrane and stuff like that, which kind of, it's a very indirect evidence, right? But at least suggesting that they somehow interact with the inside and the outside of the cell.
Right.
So,
yeah.
So the point is, right, we don't only see them inside of the cell.
Well, some of those, metabolosome type structures are for very ancient biochemical pathways too. So one would predict that there would be a wide variety of metabolosome, something not quite organelle, or maybe we want to flirt with the idea of a burgeoning organelle or a metabolic organelle. So that's just something, interesting to think about. Viruses aren't everything. I would be really excited about some, structured metabolic pathways, for instance, evidence of that in these organisms.
So one thing that we're doing right now that is ongoing, I think it will be on the cryo-ET next week, is that we will do cryo-ET, not only on one time point, but hopefully in the future also on more time points. So for example, do we see that these structures are only there in the log phase, or do they go, or are they more enriched in the stat phase, which would be coinciding with viral predation, for example. And in addition, we also do bottom-up proteomics to identify, mainly for the identifying what these green things are, because they are everywhere. They make up a large amount of the cell, and we have subtomogram images basically where they show they have basically a quasi-crystalline structure. I have no idea what it is, but it's really weird. But whatever these things are really big. This cell is 800 nanometers. These things are really big. So the idea is that we do fractionation of a protein, so to enrich large protein structures, and we should see that in the bottom-up proteomics what it is then. Yeah, and then we also we are working on, at the moment, just for MCR, but in the future do antibody staining of different proteins as well. So we do synthetic antibody, raising synthetic antibodies, basically. So this is all work that could be done by the future postdocs and grad students who want to join the lab, right? So that's a treasure trove, really, right? So once you have a culture, there's just so much you can do, basically, right? Yeah.
Very exciting, thanks.
Thanks a lot for the suggestion, Nicole, I appreciate it.
Okay, so now let's go into move on and do some networking. Let's go to the breakout groups. Thank you, Roland, for your talk.
Thanks.
Just a moment. Yes.